Hello, in this video we're going to review metabolism, or how cells get energy. Remember, there's two processes, respiration and photosynthesis, so we're going to go over both of them very briefly in this review video. I've got a trusty partner, co-teacher here, Landon, so he'll be here with me talking about these things. So first of all, we talked about two different types of pathways. One is called catabolic and one is called anabolic. If you remember, catabolic is the process of going from big to small. So breaking down big compounds, like this is glucose and oxygen, and breaking it down to smaller compounds like water and carbon dioxide to make ATP. Anabolic is the reverse. It's basically going from smaller pieces like water and carbon dioxide, adding energy to create big molecules like glucose. So this one represents respiration. Respiration is catabolic, and anabolic is photosynthesis. So one of the concepts that the AP Bio exam tells you that you need to know is you need to know that ATP powers cellular work by basically coupling or doing both exergonic and endergonic. If you remember, if I go down here, exergonic, X meaning out, basically releases out energy, and endergonic, basically absorbing energy. The cell does three kinds of work. You should know: mechanical, transport, and chemical. So the cell does these three things. But to do work, to be able to do this, you need to get energy. And I don't know if you remember Mr. Shi talking about this, but this fancy word called coupling. Coupling means basically to use energy to get energy. His example was basically you have to burn energy to walk to Zabin, to the cafeteria, to eat. So that's the process of getting rid of energy to create more. So essentially, cells have to energy release or do exergonic reactions to do an endergonic reaction. Another concept you need to know is the term enzymes. You should know enzymes is a fancy word for things that speed up a reaction, but do so without requiring energy. Another name for an enzyme is a catalyst. A catalyst is a chemical agent that speeds up a reaction without being used in the reaction. So an enzyme is an example of a catalyst. Hydrolysis, which we talked about in the last video. Hydrolysis, you mean using water to break down something. So if you break down sucrose is a sugar, with the enzyme sucrase, you break it down even faster than if you didn't have sucrase. So how does an enzyme work? So this big blue thing you see on the side here, this is an example of an enzyme. And this red thing is the example of a substrate. You should remember that an enzyme kind of goes into the substrate kind of goes into the enzyme, and the place where it goes is called the active site. Another substrate usually comes in and it connects the two, and the enzyme kind of puts them together. Or in reverse, maybe the enzyme takes two things, they sit on the active site, and then it breaks them apart. But that's what an enzyme is. And enzymes are very specific. They only work for specific things. So the last slide, we talked about sucrase. Sucrase only works with sucrose. So enzymes are very specific. Here's an example of a reaction. So, for example, you have these reactants or something that you're going to do something to. You add some energy, so it's in this transition state, and then the energy decreases for you to get the products. So the exact same thing, but this is the difference between with an enzyme and without an enzyme. So if you do it with without an enzyme, you require more energy. You have to go all the way up here and eventually to get down to where the products are. But if you had an enzyme, you don't need as much energy to do something. It's kind of like using a car to get places. You could obviously walk somewhere and it requires more energy from you. Or if you had a car, you could still get to the same spot, but not use as much energy as if you had to walk. Some more little vocabulary words you need to know. Cofactors. These are non-proteins. They're enzymes, and they're helpers also. They're called cofactors. And coenzymes, or organic cofactors, such as vitamins. So vitamins and minerals, things that you eat, they help you digest or get the nutrients that you need, but they are not necessarily as important as traditional enzymes. So for example, this is a normal binding. Oh, so those are cofactors. So cofactors are things that help enzymes, help catalysts, but they're not the primary helpers. This bottom part of the slide, we spent a lot of time, Mr. Shi spent a lot of time talking about something called inhibitors. Inhibitors are things that stop the enzyme from doing its job. Inhibit means to stop. So normally, here's a substrate, and it goes into this enzyme, this protein, and sits in the active site. It looks like Tetris, it fits exactly, and it does its job on the substrate. But if you had inhibitors, for example, this competitive inhibitor, all of a sudden the substrate can't do anything, can't go into the enzyme. That's called competitive inhibitor. That's different than a non-competitive inhibitor. What happens is that this thing kind of pushes the enzyme at the other side, and all of a sudden this space, this active site where the substrate's supposed to fit, it doesn't fit anymore. So that's the difference between competitive and non-competitive inhibitors. We also spent a lot of time talking about allosteric regulation. Spent like 2-3 classes talking about this. So these are what they call allosteric activators and inhibitors. So what happens is that a protein's function at one site is affected by the regulatory molecule at another site. This may inhibit or stimulate, this may stop or speed up the enzyme's activity. So here's an example. Here is a allosteric enzyme, and it has 4 pieces. Now, if it has this activator, for example, this green thing happens to sit there, it'll work. Like, basically, it's like the key that makes all these things work. On the contrary, if it got this red thing, which is called an inhibitor, then it cannot work. It's inactive. And so we're saying, like, why would I want something to be inactive? Shouldn't everything always want to be active? Well, maybe your body or your cells do the job that it needs to and doesn't want to keep doing it. For example, in your stomach, you want to break down things, but you want to keep breaking down until you have no more stomach. You want to stop at a certain point. So that's why these enzymes, they switch between activators or inhibitors. Another type of allosteric activation is cooperativity. In this case, the substrate kind of... so this thing is inactive, but when the substrate comes in, the rest of it switches to an active and more stabilized form. We talked about feedback inhibition. So this is a type of inhibition. We just talked about inhibition, and how it works is, for example, you have a substrate. This one's called 309, and it sits on the active site. When it sits on the active site, this enzyme changes form, and it changes form again, it changes form again, it changes form again, and eventually this enzyme turns what was once 309 into something called isoleucine. And what happens is that this isoleucine, the thing that it creates, is actually an inhibitor. So this thing can go back and sit on the original protein, the original enzyme that fixed it, and it sits at the back or in the bottom. And what happens is that it doesn't allow this enzyme to work anymore. So you're probably wondering again, why would I make something that would destroy itself? Basically, it's a pathway that allows the enzyme, or tells the enzyme, you don't need to work anymore. So like it keeps making this, it keeps making this, and keeps making this. And when it's done making it, it creates a product, which is essentially a key that tells it to shut off now. We don't need you anymore. We don't have to work anymore. So long story short, this enzyme, if it doesn't get this, will just keep on working. It'll keep on going, keep on working. And if it doesn't work, or if it keeps on working and it doesn't need to do the job anymore, it creates this to sit in the bottom, which squeezes my active site, and nothing can go inside of it anymore. So then we talked about the basics of energy. So remember, electrons fall from organic molecules like glucose. So we basically take all the energy from electrons. If you remember when you did your stop motion videos, the main thing is that we want to take energy from electrons. We take the electrons from the food. Those electrons, bless you. The electrons are carried by NADH, which goes to the electron transport chain, and then oxygen, if you remember, takes it away because it's shriveled up and used, and we don't want those electrons anymore. So remember, NAD was the electron acceptor. NAD takes the electrons and turns into NADH and stores the electron. And it takes it to the electron transport chain. If you remember, the electron transport chain is this fancy conveyor belt that is very effective in stealing energy from electrons. And then at the end, the electron is taken by oxygen and is taken away by H2O. So here is again, I'm not going to go over this in detail, but here were the 3 steps. You remember you did videos on these. We had glycolysis, which was glucose turned into pyruvate. The pyruvate was taken by acetyl-CoA and it went to the Krebs cycle. Both of these 2 processes gave up, or NADHs, they gave up electrons to these NADHs to carry it onto the electron transport chain. The Krebs cycle also had NADH and another electron carrier called FADH2. And they all go to the electron transport chain, where it takes all the electrons and creates more ATP. So if you remember, glycolysis only took 2 ATP, Krebs cycle only created 2 ATP, but the electron transport chain and oxidative phosphorylation created 34 ATP, and that's where all the energy is made in respiration. Per glucose, per unit of small simple sugars, you get 38 ATP. Here is the mitochondria. If you remember, all this takes place in the mitochondria. The glycolysis takes place in the cell, takes place in the cytoplasm. But then when it goes into the citric acid cycle, it goes in what they call the matrix. And the matrix is the inside of the mitochondria. So you may be saying, like, what's inside of the inside? So it has two membranes, basically it's wearing two shirts. It has two protective covers. And then this actual membrane, that's where the electron transport chain takes place. If you remember, glycolysis usually takes place with us in the presence of oxygen. So if we have oxygen, we do respiration, which means we break down food. Remember, oxygen is necessary because oxygen takes away the electrons at the end, and it makes all this stuff. But there are times, whether it's us or other organisms, where they don't have the oxygen to take away the electrons at the end. So they do something called fermentation. This occurs in plants and animals. This occurs in the cytosol. And what it does, it just repeats glycolysis over and over again. Obviously, you don't create a lot of energy because you get all the energy in ETC. But in fermentation, you only do glycolysis. The great thing about it, and the reason why you don't care about glycolysis, is because you don't need oxygen. In some places, you don't need oxygen. And this creates alcohol and lactic acid. So in your body, like say for example, you're going to exercise, you're going to the gym, you're going for a run, and you're running and you don't have enough oxygen, like you're constantly breathing, it goes through something called fermentation, and it builds up lactic acid to give you energy in your body. We also do this for whether it's root beer or fermentation of real beverages that we drink. That's how they create it also. They do it in a place where there's no oxygen. Two types of fermentation, we talked about these. Alcohol fermentation, you take the pyruvate at the end of glycolysis and it creates ethanol. And this is bacteria and yeast, and they do this for brewing, winemaking, and baking. Lactic acid, which is what we do in our muscles, we take the pyruvate and they turn it into lactate. And they use this also in fungi and bacteria to make cheese, yogurt, etc. Just a quick idea or quick note that I told you guys when we taught this to you the first time, lactate buildup does not cause muscle fatigue and pain. So some of you are saying, like, oh, if I'm out of energy or I'm out of oxygen and I only do fermentation, this is bad for my body. Well, they used to say that, but it does not cause muscle fatigue or pain. It basically helps you get energy when you don't have enough air. And the whole point of this, why you do fermentation, is because we can't do Krebs cycle, we can't do electron transport chain, and doing fermentation gives us more NADs, and which allows us to keep doing glycolysis over and over and over again. Where do we get our fuel? Where do we get our energy? The predominant energy comes from carbohydrates. So when we talk about glycolysis, Krebs cycle, etc., that usually comes from carbohydrates. Fats also help with that. So glycerol and fatty acids, they also can do that. And the same thing with proteins. But the majority of our energy comes from sugars and carbohydrates. And depending on what you are, like if you're a fat or if you're a protein, you enter this respiration at different points. So unless you're tired of seeing this chart, which we did before, energy goes through glycolysis, and then which is respiration, glycolysis, the Krebs cycle, and then electron transport chain. If you don't have oxygen, that's called anaerobic. You go through fermentation, which is either lactic acid to create lactic acid or ethanol, again, so you can keep doing glycolysis. And then the last thing for this video, we're going to talk about photosynthesis. This was one of the few things that, or one of the last things we talked about in this year. Remember mesophyll, which is chloroplasts in the cell of a leaf, and stomata, which is the pores in the leaf. These work together to get carbon dioxide and to do photosynthesis. And then we talked very briefly about chlorophyll. Remember, these things are called thylakoids, and this whole thing is called a chloroplast, and the stacks of these are called granum, and these little individual stacks are called thylakoids. So remember, respiration was 3 processes, photosynthesis only 2. The light reactions, which is photo, and Calvin cycle, which is synthesis. If you remember, light goes into the light reactions, which goes into, remember, this is weird, it starts with photosystem 2. It excites the electrons from water, that's why you have to water plants, and that excitement of the electrons from water helps generate ATP and helps power this Calvin cycle. Once you excite the electrons and use all its energy, it leaves as oxygen. That's the oxygen that we breathe. And then it all goes into the Calvin cycle. And at the end of the Calvin cycle, it makes sugars that plants can use and save later. Sorry, Landon wanted to stand. So another view of the light reactions. If you remember, light excites electrons from water, and it goes through photosystem 2, and it goes through an electron transport chain. So once the water's electrons are excited by light, it goes through an electron transport chain, which is here. And then it goes into photosystem 1, where those electrons that were just went down the electron transport chain, they're re-energized like a rechargeable battery by light. And again, this is called photosystem 1. And it goes through a mini electron transport chain. In this case, you don't make ATP like the real electron transport chain. What you make is NADPH and ATP to go into the Calvin cycle. Remember this chart that helps you remember how the light reactions work. A photon of light gives energy to an electron, then it goes down an electron transport chain to make ATP. It's recharged by another photon of light in photosystem 1, and the electron gets carried by NADPH. Both respiration and photosynthesis use something called chemiosmosis to generate ATP. Chemiosmosis, if you remember, is a fancy term for this process right here. You remember in terms of how to make ATP or how an electron transport chain works, it basically, those electron energy is used to push H plus out of the membrane, and then when the H pluses go back in the membrane, it powers a turbine, and that turbine connects DP and P to create ATP. So I know it sounds confusing, but I hope you remember that chemiosmosis, or this part of the electron transport chain, how you make energy is basically by pushing H plus ions out, and when they come in, it spins and creates energy to create ATP, and that process is called chemiosmosis. The Calvin cycle, again, it's kind of hard to remember all of this. It's been a while as well, but again, I will tell you very briefly that, remember, you get in carbon dioxide, and you have a molecule called Rubisco. What Rubisco does is it comes in and it's being fixed. So remember, carbon fixation means to fix the carbon. It rearranges it and makes the way that it can be used. So the Rubisco, the very first process is that it's being fixed. When you fix it and you add energy and you take the electrons from the NADPH, it reduces into something called G3P. So this was once 6 carbons, and it's now a 3 carbon called G3P. You may be wondering what G3P is, or maybe you don't even care what G3P is, but G3P is just one piece of sugar, or half of a sugar. It's called a 3C sugar. It could become sucrose, it could become starch, it could be a lot of things, but it only makes a piece of it. The rest of it gets regenerated by ATP, and it becomes Rubisco again, and that's why they call it the Calvin cycle, and you start over and over again. And then when it goes through a second turn, when it does this a second time, then you get a real sugar, but only one turn makes just half a sugar. It has to go through it twice to make a full sugar. Photorespiration, we talked about respiration and photosynthesis a little bit different depending on what kind of plants you are. You may remember we talked about 3 different kind of plants: C3, C4, and CAM. C3 plants, they're the ones that you know, like rice, wheat, soybeans, and it goes through photosynthesis like I taught you, like the Calvin cycle, and then you have Rubisco, and it goes in the mesophyll cells. But depending on the different kinds of plants or where you are, there's different types of ways you do photosynthesis. For example, if you're a sugarcane or you're a grass, you do something called, or you're called C4, or a C4 plant. What happens is that carbon fixation and the Calvin cycle, they happen in different cells. So because sugarcane and grass, it tends to be a little bit hotter, what happens is that they split those 2 processes so that you maximize the creation of sugars. That's different than CAM. CAM is like cactus or pineapple, and we're talking about places that are really, really hot, or the temperature is really hot. The problem is that you can't do photosynthesis when it's super hot, because you have to open up your pores, and when you open up your pores, you lose water, and plants don't like that. So in CAM plants, they do carbon fixation and the Calvin cycle at different times. So like at nighttime, when it's not so hot, that's when they do all these things. Instead of Rubisco, C4 uses PEP carboxylase, and CAM uses organic acid. At night, that's when they do carbon fixation, and during the day, that's when they do the Calvin cycle. For C4, so let's get started. Mesophyll, they fix the carbon dioxide, and the bundle sheath, the second part, does the Calvin cycle. So again, C3, like the normal plants that we know, they do everything in the mesophyll cells. C4 plants, one part does the carbon fixation and another part does the Calvin cycle. And CAN plants, they're the ones that are in the deserts. That's why they have to fix carbon dioxide at night, so that when they open up their pores to get carbon dioxide, they don't dry out. And then during the day, that's when they do the Calvin cycle. So just to wrap up, you should know the difference between respiration and photosynthesis. Remember, they're reverse. Most people think respiration is animals and photosynthesis is plants. True, but respiration can also be done by plants. You need oxygen and food, and you produce carbon dioxide. Photosynthesis is the reverse. It produces oxygen, and that's why we breathe out carbon dioxide, they breathe out oxygen, and we help each other out. Respiration is in the mitochondria, photosynthesis in chloroplasts. Then we do oxidative phosphorylation, and photosynthesis is photorespiration. And then just like for the both of them, we talked about oxidative phosphorylation, we talked about chemiosmosis, where the H plus protons go up and outside and come back in and power to create ATP, and that happens in both processes. So that's it for this video. Hope you guys enjoyed. Hope you're okay to land in, but hope you understand this review, and I'll see y'all for the next review video.